Okay, so enzyme introduction. Now that we discussed how proteins are made, we're going to go into one of the really important functions of proteins, which are enzymes. So an enzyme is a biological catalyst that is not used up in the reaction. Enzymes work kinetically, not thermodynamically. So this means that enzymes do not affect the delta G of the reaction, but lower the activation of the energy. Lower the activation energy of the reaction to speed it up, that should say. So what that means is a reaction is going to proceed towards its equilibrium, and when you add an enzyme, it is not going to affect the equilibrium at all. It's going to affect how quickly the reaction will proceed to that equilibrium. So it's not affecting the delta G, it's not affecting the energy of it, it's just affecting the speed and the rate of the reaction. So that's why we say it works kinetically, not thermodynamically. It's not changing the thermodynamics of the reactants or the products. Enzymes are categorized into different classes based on their function. So if you look to the right, these are 6 major classes. So we have oxidoreductases, transferases, isomerases, hydrolases, lyases, and ligases. So you can pause, download the slides, just take a look at that. That's a pretty good graph. Definitely be familiar with what they do. And it's also important to keep in mind that just kind of like everything else we've been talking about, you'll see a bunch of different classifications for different like when you're going over different topics or when you're getting it from different sources. So like enzymes, for example, you may also see them categorized as kinases, polymerases, phosphatases, phosphorylases, and proteases. So it's just important to understand what the enzymes actually do, because those other categories, depending on who creates the graphic or who you're talking to, those will all fit into one of the categories that I have to the right. So I would say definitely at least these 6 major classes you should know, and those that I listed below as well, you should definitely know what they do. So enzyme structure and function. Enzymes fold into specific formations that always have an active site. This is the location of enzymatic activity. So there are 2 models that seek to explain the interactions between substrate and active site. So the substrate is obviously the molecule on which the active site is acting. So the lock and key model, or the active site model, claims that the substrate and active site are a perfect complementary shape and fit together, similar to a lock and key. So they are completely complementary shapes. The substrate goes into the active site just like a key would fit into a lock. The reaction occurs, and then the reactants are released. That differs from the induced fit model, which claims that the active site and substrate have slightly different shapes, but the enzyme undergoes a conformational change when they bind. So it actually changes to fit into the form of the substrate. In both models, the enzyme lowers the activation energy by stabilizing the transition state. So we know for a fact that that's how enzymes work. And to the right, this is a graph that you'll definitely see pretty often. So if you see this larger hump, that's the uncatalyzed reaction. And in order to start that reaction, we have to get from this reactant point all the way up to the transition state. So if you look at the catalyzed reaction, we have to go a lot. We don't have to go as high as the uncatalyzed reaction because the activation energy is much lower. So it's going to be much easier to get over that lower hump than it is to get over the higher hump. And that's basically what enzymes do. They just lower the activation energy, but you can see that in both reactions, the energy of the reactants and the energy of the products are both exactly the same. Right over here, this free energy change is exactly the same in both reactions. And that's what we were talking about before, how it doesn't change that delta G. It just changes the kinetics. It gets there a little bit quicker. So regulation of enzymatic activity. Many enzymes do not have to be constitutively active. Constitutively active means that they're constantly being produced and constantly being created. And there are multiple ways that they can be regulated. So some enzymes can be modified by covalent bonding. Enzymes can be activated or deactivated by adding a phosphate group, for example. Some are produced in an inactive form, which is called the zymogen, and they must be cleaved in order to become active. So sometimes they'll have a cap or some sort of inhibiting protein that's attached to them. And when you cut that off, the enzyme becomes active. So other enzymes can only begin catalytic activity by associating with a regulatory peptide. So they have to associate with a certain protein before they become active. And then finally, some enzymes are regulated allosterically, meaning modification of activity occurs at a site other than the active site. So that could be, either inhibition, or it could be like excitatory, and it could encourage the enzyme activity. Quantifying enzymatic activity. Enzyme kinetics is the study of the rate of formation of products from substrates in the presence of an enzyme. Before we look at a graph, let's discuss some important variables. This stuff is very important. I always see this on MCAT. Michaelis-Menten plots, enzyme kinetics, Lineweaver-Burke plots, which we're going to spend a little bit talking about right now. Definitely know this stuff. If this is something that you struggle to keep in your mind, it is definitely worth devoting the time to, because this is very high yield. Enzyme kinetics is very high yield on MCAT. So let's just go over the variables. V stands for the reaction rate, or the amount of product that is formed over time. It's usually measured in moles per second. So V comes from velocity, obviously. But the rate that we are measuring in terms of enzymes is measured in moles per second. And that's moles per second of product that's formed. So that S in brackets is the substrate concentration, obviously. Vmax is the rate of the reaction when all enzyme active sites are saturated. When adding more substrate does not increase the rate of reaction. So if you only have a set amount of enzymes, you're going to add substrates, and it's going to increase the rate up until a certain point, because at a certain point, when you add substrates, there's not going to be enough enzymes to turn all of those substrates at a higher rate. So we reach that saturation point, and that rate we refer to as Vmax. Km is also an important variable, and it's referred to as the Michaelis constant. So it is the substrate concentration at which the reaction is proceeding at half of its maximum rate. So Vmax is a rate, Km is a concentration, and they're related. We can use the X and Y axis to relate that. So if we look at Vmax, we slide all the way over to this y-axis. We want to look at half of Vmax, so we find halfway between, and then we just go over to the graph so we can find the corresponding X point. Because that X is going to be Km, because that is the concentration. Since concentration is on the x-axis, that is the concentration at which we are at half of Vmax on the y-axis. So that's the concentration. If Km is low, that means you only need a small amount of substrate to saturate the reaction, or the enzyme has a high affinity for that substrate. So Km and affinity are inverse. They're inversely related. The lower the Km, the higher the affinity. The higher the Km, the lower affinity. Something to keep in mind. Enzyme cooperativity. So when enzymes form multi-subunit complexes, or they have multiple active sites on a single polypeptide, they may behave according to cooperativity. So in positive cooperativity, the binding of a substrate to one subunit increases the other subunit's affinity for the substrate. So remember the tense and relaxed states of hemoglobin. Again, hemoglobin is not an enzyme, but when it comes to cooperativity, it's a good example to look at, because it's something that we learn about in other topics. So that tense state, it's a little bit more difficult to bind, for the substrate to bind, but then once one of the subunits binds to that substrate, it enters a relaxed state where it's a lot easier for the other subunits to bind. So that's what positive cooperativity is. It's after that first binding, the enzyme actually finds it easier to bind with the substrates. And then negative cooperativity is the exact opposite. That is much less common on the MCAT, but I mean, if you understand what positive cooperativity is, you understand what negative cooperativity is. And you really only need to recognize the graph for positive cooperativity. So if you look right over here, this is the Michaelis-Menten plot for a positively cooperative enzyme. And this has a sigmoidal structure. This has a sigmoidal curve right over here. This S is called the sigmoidal curve. So if you look at it, the reason why is because we start in this tense state over here. So the increasing rate is slow because we're in that tense state. But as we add more substrate, because of positive cooperativity, that enzyme is going to increase its affinity for the substrate, and it's going to start binding a lot more quickly. And that's why we see this steep increase. And then obviously we're going to hit our Vmax just like any other reaction. So it levels off right at that Vmax. And that's pretty much why we have this sigmoidal curve for positive cooperativity. Okay, so Lineweaver-Burke plots use the same variables as the Michaelis-Menten plot, but they're graphed using a different formula. So the formula is not super important to memorize, but if you're confident in your ability to memorize formulas, it's a pretty good one to know, just because we're going to talk about a bunch of variables below. And if you ever forget which variables stand for what, you can always derive it from this equation. So you don't have to know it, but I mean, if memorizing your formulas is memorizing formulas is your thing, and you think that's going to help you, then I say go for it. So 1 over V equals Km over Vmax times 1 over substrate concentration plus 1 over Vmax. And the reason why we put it in this form is because the formula allows us to analyze the enzyme kinetics linearly. So if you look at that formula, it is in the form of y equals mx plus b. So if we look below, we can analyze that that formula, and we can find all of these, we can find a lot of information from the Lineweaver-Burke plot about this interaction that we wouldn't be able to find on the Michaelis-Menten plot. So the Michaelis-Menten plot is very good to look at visually because it shows us, where it levels off at Vmax, but the Lineweaver-Burke plot is very good for quantitative, getting quantitative information from the actual reaction. So our x-axis, because of this formula, is going to be 1 over the concentration of the substrate, and the y-axis is going to be 1 over the speed, the reaction rate. So the slope of our graph is going to be Km over Vmax. The y-intercept is going to be 1 over Vmax, and the x-intercept is going to be -1 over Km. And you should definitely know those formulas for the MCAT.